Hello friends, if you are preparing for ASRB, NET or ARS mains, ICAR, JRF, SRF, PhD entrances or agriculture officer exams, this topic is extremely important because it has been recently reported in India and is becoming an emerging disease of rice, the southern rice black-streaked dwarf virus. Before understanding the disease, let's discuss about the history part. Many students actually confuse between this rice black-streaked dwarf virus and southern rice black-streaked dwarf virus. These are different viruses. I will explain the difference in this video, okay? So without further delay, let's start the video. The rice black-streaked dwarf virus, this was first reported in Japan 1950s. But the southern rice black-streaked dwarf virus first reported in China 2001. The major epidemic actually seen in China and Vietnam 2009-10 with 30 to 50% yield loss in severely affected field. In Indian condition, among these two, the rice black-streaked dwarf virus and southern rice black-streaked dwarf virus. This southern rice black-streaked dwarf virus was confirmed. It was reported in India, the SRBSDV. Remember this. So, which black-streaked dwarf virus was reported? Southern rice black-streaked dwarf virus was already reported in India, in Punjab, Haryana, and Uttarakhand during Kharif 2022. And fresh outbreak was reported in Kharif 2025 in Punjab and Haryana. So, this is about the history and current status of the disease. Then about the symptoms. So both this RBSDV as well as SRBSDV both produce similar symptom. From the name only you can predict the virus symptom. See, in most of the viruses, stunting, yellowing, these are common. Okay. Here also the name only suggests that this is rice black-streaked dwarf virus. So first thing is the virus produce black streaks. So where the black streaks are produced? So black streaks are produced on the leaf veins as well as stems. Here you can see in the photo the black streaks are there. Okay. So it produces black streaks on vein and stem. Then dwarf virus means it causing stunting as well as dwarfing of plant. The height of the plant will substantially decreased. Okay, and another characteristic symptom is dark green leaf. If you see the field from distant, you feel like it's a very healthy field, very healthy plants are there because the plant leaves become very dark green. Okay, excessive tillering was seen. But if you go to close for a closer view, if you go into the field, then you can get this black strips, poor root development, delayed or no panicle emergence, poor grain filling. So these are the symptoms for both the RBSDV as well as SRBSDV. Okay, similar symptoms are there. The major difference lies in the vectors. So we will discuss in the next slide. Next, the causal organism. So black-streaked dwarf disease was caused by black-streaked dwarf virus, and southern rice black-streaked dwarf virus causing the southern rice black-streaked dwarf disease. The morphology of the virus is very similar. Both are icosahedral non-enveloped viruses with diameter of 70 to 80 nm. Icosahedral. This is a type of symmetry for the specifically for the spherical or polyhedral viruses. I have already discussed in my previous video in plant virology series that the viruses is showing three types of symmetry. One is helical symmetry. Helical means either it is a flexible or rigid rod. Then second important symmetry is the icosahedral symmetry for the spherical viruses, and third important symmetry is bacilliform symmetry for the bullet-shaped viruses. Then the family for the Rice Black-Streaked Dwarf Virus or SRBSDV is Spinareoviridae. It is previously classified under Reoviridae, then reclassified as Spinareoviridae, and genus Visivirus. The genome is double-stranded RNA and it is 10 segments. So the genome is segmented. It is multipartite virus. It is not seed transmitted. Then how it is transmitted? It is transmitted by insect, specifically the plant hoppers. Now the interesting thing is two different insect transmitting these two vectors, these two viruses. The major virus, that is the southern rice dwarf virus, that is already reported in India. That principal vector for that virus is WBPH, white-backed plant hopper. The scientific name is Sogatella fucifera. Remember both the common name as well as the scientific name. This is mostly asked question. Okay, ASRB favorite question. If you appear in an interview also, definitely they will ask if you are working in virus, or if in general also for transmission part they may ask. Okay, so remember both the common name as well as scientific name. Whereas for the RBSDV, the vector is small brown plant hopper and the scientific name is Laudelfax striatellus. Okay, so remember the scientific name for WBPH as well as small BPH. And the transmission mode is persistent propagative transmission. See, the insect transmit the virus in three manner: non-persistent, then semi-persistent and persistent type. Here the virus is transmitted in persistent manner, means the vector remain viruliferous lifelong. Okay, and it is propagative in nature. So persistent type is categorized into two groups: circulative, propagative. So in case of circulative type, the virus will circulate in the hemolymph of the insect, whereas in case of propagative type, the virus will multiply. Okay, virus will multiply specifically in the salivary gland of the insect. So virus multiply inside the vector. Both the nymph as well as adult transmit the vector. This is also very important. See, these type of question only asked in assessment reasoning type. So 2-3 option will be given. You will get, means in multiple choice also you can get this type of question, means whether nymph is viruliferous or the adult, who will transmit the disease. Then this vector is transmitting the virus in a persistent propagative or persistent circulative manner, the scientific name of the vector. So this is very important slide. Okay, don't skip any single point from this. Remember all these important points. Next, about the disease cycle. So primary source of inoculum or reservoir for the virus is the infected rice plant, obviously, then volunteer rice plant, grassy weeds, okay, an alternate host for the hoppers. So this transmission is carried out by the viruliferous plant hoppers. Inside that, the virus also multiply and circulate inside the body, and once the vector become viruliferous, it spread the virus to multiple plants. And major important thing is the early infection at seedling stage particularly cause maximum damage. Okay, so the spread occur through the plant hopper, and primary source are these things: the infected rice, volunteer rice, and weeds. Next, the favorable condition. See, for most of the viral diseases, the favorable condition is warm, humid, and cloudy weather. And if it is transmitted by the vector, then definitely high vector population will be favorable. Early stage is also favorable. Then continuous rice cultivation, presence of the primary source of inoculum, that are grassy weed as well as alternate host. Okay, these are the favorable condition for the epidemic development. And remember this point: high vector population is directly proportional to high disease incidence, because this is spread by the WBPH. Next, the management practices. For the viral diseases, IDM was most preferably used, the integrated disease management. Why? Because for the viral diseases, no such resistant variety available, no such chemicals available. Because for bacterial disease, you can directly apply antibiotics or bactericides. For fungal disease, you can directly apply some fungicide, but here the viricides are not available. So concern we should major thing that we should consider is use of healthy plants. The planting material should be free of disease or free of viruses. Then if it is transmitted by any vectors, then we have to control the vector population. So raise the nursery bed under insect proof net and monitor and control the plant hopper population. Roguing is very important in case of viral diseases. Then removal of grassy as well as volunteer rice. Then adjust sowing time so that it avoid the peak vector population. Then growing of tolerant varieties, because resistant varieties are mostly not available, so grow tolerant varieties. So these are the management practices for the SRBSDV. Next, the major challenge I have already told. The major challenge is unavailability of the viricides. Then the insect, because the vectors are highly mobile, and major here in case of SRBSDV, the persistent nature of virus. Once the virus is once the virus enter into the system of the insect, the virus will remain in means the insect will become viruliferaous throughout its life, both the nymph as well as the adult. So it will become very dangerous for our plants. Then lack of resistant variety, insecticide limitation. See, this sucking pest, the hoppers, this so very high level of resistance. The emergence of resistance biotypes are very frequent. So we have to use insecticide in very limitation way. Okay, so another thing is use of or proper use of insecticide is also we have to major concern. Then lack of awareness and timely monitoring. We have to monitor the disease as well as the insect pest population also, the vector population also. So these are the challenges or difficulties that we have to face for the effective management of SRBSDV or Southern Rice Black-Streaked Dwarf Virus. So this is all about Southern Rice Black-Streaked Dwarf Virus, about the history, where it is originated. Then fresh outbreak occurred in Kharif 2025 in Haryana, Punjab. Remember this, where it was first reported in India. Next, about the major symptom, the dark green leaves as well as black streaks, and also the stunting. Okay, about the causal organism, it is 10 segmented double standard RNA virus, spherical virus. Okay, next about the vector, this is most important, the plant hopper. Then about the management practices. So I hope it is clear to you all people? Okay, so if you found this video helpful for you, please like the video and subscribe my channel Plant Pathology Pathsala. Thank you.